Everybody knows. You flip a coin, 50-50, heads or tails. When you flip the coin, it's a 50% chance you're going to get heads, 50% chance you're going to get tails. What's also interesting about this idea of 50-50 is we can project this. We can use this as a means to discuss genetics. If you have two genes, or let's say one gene. One gene that's going to be in the sperm, one gene that's also going to be in the egg. Sperm coming from daddy. For one character, we know there's going to be two copies, because remember, daddy's diploid. Due to meiosis, which of those versions are going to be the one that ends up in the sperm? 50-50. Which one will make it? Same thing with the egg. For every gene, there's going to be two versions. Thanks to what we know about meiosis, that egg's going to divide and divide again, and each of those resulting eggs from the meiosis, each of those cells, which version will be there? You look at the figure to the left here. Two coins, going to be flipped. Big R for heads, little r for tails. We know you flip one, it's 50-50 that you're going to get heads. It's a 1 in 2 chance. That's all you're going to get, heads or tails, 50-50, one or the other. Same with the second coin. But what happens when you go and you try to calculate flipping them both? Well, spoiler alert, you look here, you see what we call a Punnett square. So if you were to sit there and say, okay, instead of the coins being flipped, boom, you're thinking of the 50-50, as it applies to sperm, as it applies to the egg. We can go through and set up these little visualizations so that we can determine the probabilities of what's going to wind up in the offspring. You go through, you set up the square. If you're only doing one gene, it's 50-50, so you have two. Big R, little r. Big R heads, little r tails. So you're going to have a two of what you can get across, two probable outcomes across the top, two probable outcomes down the side. Then you just fill in the columns and you fill in the rows. Notice up at the top left, right up here, right up here, big R, big R. That means what's the probability if I flip both coins that I'm going to get both heads? Well, turns out it's 1/2 times 1/2, or 1 out of the 4 squares. What's the probability I flip both coins and I end up with two tails, two little r's? Again, 1 square out of the 4, or 1/2 times 1/2. What's the probability that if I flip two coins, at least one of them will have heads? Well, that's 1, 2, 3. 1/2 times, or I'm sorry, yeah, 3/4. 1/2 plus 1/4. We can apply this out even further. These probabilities are ways for us to calculate the chances of what traits will be evident in offspring. We can do it with 1 trait, 2, 3, 4. It's just the squares, the Punnett square, which we'll talk more about in a later lecture, just get bigger and bigger and bigger with the more probable outcomes. Scientists, post-Mendel, came up with a couple probability rules. The multiplication rule says that if you want to know the probability of A and B occurring, you get both heads and you get 1 coin tails. You multiply the 2 probabilities. 1/2 times 1/2 gave us the 1/4. The sum rule, well, what's the probability of A or B occurring? Add the probabilities up. The 1/2 plus the 1/4 gave us the 3/4. We can go through and we can discuss this in terms of, 1, 2, 3 characteristics, 3 genes. If you look over here, you're going to take some organism, 2 genes, Y and R, Y, big Y, big R, little r, and you're going to mate them together. What's the probability of the offspring having big Y, big Y, big R as the 4 alleles for the 2 genes present? Well, 1/4 times 1/4, a 1 in 16 chance. Now you can sit there and go through and do the probabilities. 1/4, big Y, goes back to our 2 coins. Our 2 coins, which, coin 1, coin 2. Go back to what, a couple of last slide, 2 slides ago, where it was, okay, 1 and, 1/4, 1/4. Multiply that throughout, 1/16. What's the chances of, big Y, little y, big R? Well, you go through and you think about that. Slide again. At least one of them being big R, or here big Y. Well, it was half. Half of the 4 possible outcomes. How many of them were big R, or heads? Only 1 out of the 4. 1/2 times 1/4 gives us 1/8. We can carry it out. Here, the same thing. If it was 3 different genes. We could do it with 4. We could do it with 5. Now, since this course is totally online and we're not really meeting, we're not going to really be doing these calculation of probabilities. If this was an in-class thing, if we had classes where we were meeting, yes, we would be going over work packets and stuff and how to do these calculations. But it's one of those things, trying to do it online and to walking you through it through these little tape things, not going to work out. I do know that in a couple of years, when you take Albie for genetics, she'll be going over probability calculations there, a little more in depth and getting you up to speed then.